Welcome students, this is the instructional video for the cell cycle instructional project. Before we begin, I want to quickly remind you of what the objectives are for this portion of the instructional project. Given a multiple choice question, the students will be able to name the specific functions of the different phases of mitosis. Given an essay question, the students will be able to explain how and why mitosis results in identical daughter cells. Keep those in mind, make sure you know them in and out before you do the assessment. Okay, let's begin. So mitosis, this is the second half of the cell cycle. Remember the first half, well, I say half, but what I really mean is part, because as we learned before, interphase is much longer than mitosis. So mitosis, this is the second part of the cell cycle. So what exactly is mitosis? What's the point? Mitosis is the process by which a cell divides one parent cell, and this divides and produces two daughter cells that are identical. This is very important for you to remember. It produces two daughter cells that are genetically identical. And mitosis is broken up into four phases. First phase is prophase. DNA condenses into chromosomes. Normally DNA is loose inside of the nucleus. And during prophase, it condenses into really tightly wound chromosomes for division. The nuclear envelope breaks down, spindle fibers attach to the centromeres. These are the proteins that connect the two sister chromatids, and they push these sister chromatids toward the center. Okay, the nuclear envelope breaks down so that the chromosomes can escape the nucleus and be out in the cytoplasm. Moving on. Metaphase. Chromosomes align on the metaphase plate. So this is important to know that this is not a specific structure. This is more like an imaginary plane right in the center of the cell. So think of it like the equator. It's not a real of the Earth, that is. It's not an exact structure. It's an imaginary line that we've drawn at the center. So as you can see here, these are the chromosomes. Spindle fibers are attached to the kinetochore proteins on the centromeres, and they are pushed to the center, and they align on the metaphase plate. The third is anaphase. Spindle fibers pull these sister chromatids apart so that you see here, these are identical, and so that way each daughter cell gets the same chromosomes. So it pulls these apart, and then they move toward the opposite poles. Lastly, we have telophase. Spindle fibers start to break down. We have the nuclear membrane formation, so that packs the chromosomes back up in the nucleus. The chromosomes begin to decondense and go more into that loose DNA structure. And then the membrane pinches together. This is called a cleavage furrow, and this is where cytokinesis occurs. Some people say it's a separate stage of mitosis, but you don't have to worry about it. Just know that the membrane pinches together, and then these cells separate, and you have two identical daughter cells. So to reiterate, I created a concept map using Creately. So we have interphase, which is kind of our start. Everything happens here, okay? And then we move into mitosis. So all of this is mitosis. We have prophase, DNA condenses, nuclear envelope breaks down, spindle fibers attach. We move on to metaphase, where the chromosomes align on the metaphase plate. We move to anaphase, where the chromosomes move toward the opposite poles. And then finally we have telophase. Nuclear envelopes reform, cleavage furrow pinches together, and we have two identical daughter cells. So from here you're going to move on to the review. That's an optional review. If you feel confident, go to the assessment portion and take the assessment. If you don't feel so confident, look through the tutorial and the video again. Look at the objectives. Look at the review videos I have posted. Thank you.